I'm Susie Jackowski, and it's indeed a privilege to be telling you about my journey through research over the last 40 years. I'm happy to say that we've arrived just recently at a potential therapy for a pediatric neurodegenerative disease. This disease is called panthenate kinase neurodegeneration, also known as Halliburton-Spatz disease. The patients exhibit symptoms very early on. It's a rare genetic disorder. It's a movement disorder, and they develop dystonia that gets progressively worse. And it primarily affects the facial and alimentary muscles and also the limbs. And they eventually die in their teenage years. My journey started when I was trying to do some metabolic labeling to study fatty acid synthesis in E. coli. And between the time I left my postdoctoral fellowship and started my first real job at St. Jude, the panthenate I needed to do the metabolic labeling was no longer commercially available. So I had to construct an E. coli genetic mutant for a precursor of panthenate because that radioactive compound was still being sold. And I learned how to label cells with radioactive beta-alanine. But in the course of doing those experiments to optimize conditions, determine concentrations, yada, yada, I discovered that panthenate kinase was the rate limiting step in coenzyme A biosynthesis. It governed the size of the total CoA pool. So we took the DNA sequence from our E. coli pan-K and searched every available EST and cDNA library that was available at the time. This is way before genomes were published and things were at the point they are now. And we searched and searched repeatedly, and we could find nothing with any homology or similarity to the E. coli pan-K sequence. So I tabled that approach for a while, worked on a few other things, and then I was fortunate to have a collaboration with a group in England who wanted to clone the panthenate kinase from Aspergillus. So that cDNA sequence from Aspergillus allowed us to find the genes in both mouse and human that encoded 3 distinct panthenate kinase genes and, well, encoded 4 different isoforms of the enzyme. So we proceeded to knock out all the 3 pan-K genes individually to find out their physiological roles in mice. And a few years after that, I was invited to a conference that was sponsored by the NBIA Disorders Association. This is a wonderful patient advocacy group that is run by the families of patients with panthenate kinase associated neurodegeneration and other related disorders that we now know about that all accumulate iron in the brain, thus NBIA, neurodegeneration with brain iron accumulation. So at that conference, I met patients, their families, and some of the very committed physicians who were managing there was no cure, there was no treatment for this disease, but managing these patients while they progressed down their road. And I came back from that conference and I thought, knowing what we know, isn't there something we could do to help provide a therapy to change the CoA levels in the brain that would overcome these disabilities? We designed an assay that looked for both activators and inhibitors of the panthenate kinase. This process took over 3 years, 1.3 grams of purified protein and over 1 million assays to come up with a chemical scaffold that we felt we could work with. And I got my friends together, a wonderful medicinal chemist, a real biochemist, a structural biologist who'd been working with us on the structure of the panthenate kinase proteins. And I handled the biology, the cultured cells and the mice. And we got together and we decided to try and develop a therapy that would be orally bioavailable, cross the blood-brain barrier, and increase coenzyme A in the brain. So we gave our best lead compound to these mice in chow, and the coenzyme A was rescued in the brain, and they got up out of their wheelchairs and walked, and they lived twice as long. And this was a wonderful result after all of this work. COA Therapeutics guided us through the preclinical studies that were needed to prepare the IND application to the FDA. And even through COVID, we got through all the experiments, all the hard work, and we improved the compound substantially. So we actually went through several iterations, again, changing the chemical structure of the molecule, seeing how it had interacted with the protein, which guided the changes that we could make. And we ended up with I think, a very good, remarkable clinical candidate that is now moving forward into the clinic. So I retired a few weeks ago. The preclinical work is done, and I will continue as an advisor for the company as we move forward and try to help the patients with panthenate kinase neurodegeneration. So thank you for your attention, and keep us in mind.